So now we are having, that is, types of enzymes. Types, and basically we are having three types of enzymes. Three types. Number one is competitive inhibition, number two is non-competitive inhibition, and number three is uncompetitive inhibition. Now we will discuss about first the competitive inhibition. Competitive inhibition. Now what is competitive inhibition? Now what are these? Basically these are the type of inhibition. What I mean by inhibition? Which are slowing up the reaction, that are called inhibitors. Enzymes are those who speed up the reaction, and inhibitors are those who slow up the reaction. So we will talk about the competitive inhibition. That these are the type of inhibition occur by the molecule where are very, we can say, similar to the substrate and bind to the active site instead of the original substrate. Suppose that we are having here, suppose that this is a enzymes, this is that one substrate, right? So we can say this is enzyme. So original substrate will not attach here except original substrate any other molecule that is similar to this, that will attach to its active site, and block the original substrate's active site, which means the original suppose this is original, it will block this side, because we can say another substrate that is similar as its active site that will attach to this side, and it will block the original active site of the original substrate. So that is called a competitive inhibition, which means we are having another enzymes that are similar to some enzymes, we can say active site, so they bind to that one active site and block the original substrate active site, that are called the competitive inhibition. Now if we take an example, we are having penicillin, right? So penicillin is basically, this is a competitive inhibitor that attached to the we can say enzymes active site, and we can say it will block that original substrate active site. Now if we talk about the non-competitive inhibition, competitive inhibition. Now what is non-competitive inhibition? The competitive inhibition is type of inhibition when a molecule binds with enzyme at any site except active site. Suppose that this is a, we can say this is a, this one active site, right? So the substrate of these are the non-competitive inhibition are those who attach with the attach with this substrate except the active site, which means this is active site, right? So it will not attach here, it will attach like from here or here or here or here. It will not attach from the active site, that are called the non-competitive, which means they will attach to any allosteric site. This is known as, except then active site, other sites are known as allosteric sites, which means they will not attach to the active site, they will attach any of the site of the enzyme, that are called the non-competitive inhibition. So that's why when they will attach, suppose that they are attaching on this side, so they will completely we can say disrupt the structure of that enzyme, which means it will do enzyme will move like this, the structure will be like this, so this will not work as a speed up the reaction, so the whole structure will got disrupt. So they are called the non-competitive inhibition, which are attached to the enzyme, except then the active site they will attach to the any allosteric site, so they are called the non-competitive inhibition. Now number 3 we are having, that is uncompetitive inhibition. Uncompetitive inhibition. Now what are uncompetitive? Basically these are also known as anti-competitive inhibition. Anti-competitive inhibition. Now what are anti-competitive inhibition. Basically it take place when an enzyme inhibitor bind only to the complex format between enzyme and substrate, which will be add towards the enzyme and substrate complex. So they are called as the uncompetitive enzyme. The type of inhibition typically occurs in the reaction with two or more substrate or product. It will not occur in one substrate or one product. It will only occur two or more than two substrate, or we can say to the product. So that was all about the types of enzyme inhibition.